Okay, so your physiology exam is just around the corner, and you need the high-yield, absolutely essential info on muscle contraction. Well, you are in the right place. We're going to cut right through the noise and focus on exactly what you need to know to ace this topic. Let's get right into it.
So here's our game plan, nice and simple. First, we're going to break down the anatomy of a muscle. Then, we'll look at the actual engine of movement. After that, we'll find the spark that gets it all started, and finally, we'll zoom out and see how smooth muscle is different. Ready?
All right, first things first, we have to understand how a muscle is built, from the big picture all the way down to the tiny molecular level. Getting this hierarchy right is absolutely foundational for everything else we're going to talk about.
Check this out. It's like a set of Russian nesting dolls. You've got the whole muscle, right? Well, that's made up of bundles called fascicles, and inside each fascicle you have the muscle fibers. Those are the actual cells. But we go even deeper. Inside each cell, you've got these little protein rods called myofibrils, and that's where the real action happens. Now, if we put one of those myofibrils under a microscope, you'd see it's made of these repeating sections. Each one of these sections, from one Z disc here to the next Z disc here, is called a sarcomere. You need to remember that word. The sarcomere is the fundamental, the basic unit of muscle contraction. And see those alternating dark and light bands? That's what gives skeletal muscle its classic striped look.
So what makes up those bands? It's really just two main characters in the story. You've got your thick filaments, which are made of a protein called myosin. See those two little heads? Think of them as tiny motors or little arms that can grab and pull. And then you have the thin filaments, which are mostly a protein called actin. You can think of actin as the rope that the myosin motors are going to pull on.
But, these myosin motors can't just grab the actin rope whenever they feel like it. The whole thing is very tightly controlled. And the gatekeeper, the guy in charge, is a protein complex called troponin, specifically a part of it called troponin C. And the C is a perfect hint because it stands for calcium. When calcium floods in, it binds to troponin C, and that acts like a key in a lock, turning on the whole process so the pulling can begin.
Okay, so we've met our main players, actin and myosin. Now let's see how they actually work together to create force. This is the engine room. This is the mechanism that powers every single move you make, from blinking to lifting weights.
This right here, this is the whole story in one sentence. It's called the sliding filament theory. And a super important point for your exam, the filaments themselves don't get any shorter. Actin doesn't shrink, myosin doesn't shrink. They simply slide past one another, kind of like sliding a telescope closed. This sliding pulls the Z discs closer together, and that's what shortens the whole sarcomere.
So, how does this sliding actually work? It happens in a four-step cycle called the cross-bridge cycle. Imagine those myosin heads are a team of rowers in a boat. Step 1, they reach out and grab the actin rope. That's formation. Step 2, they pull. That's the power stroke. Step 3, a new molecule of ATP comes along and makes them let go of the rope. That's detachment. And step 4, they use the energy from that ATP to reset and get ready for the next pull. It just repeats, grab, pull, release, reset, over and over.
Now that whole rowing action we just talked about, well, it leads directly to this. This is a classic graph you're absolutely going to see on your exam. It shows that the amount of force a muscle can generate depends on its starting length. And as you can plainly see, there's a sweet spot right in the middle where the muscle is strongest.
And this just explains why that sweet spot exists. It makes perfect sense. If the muscle is too short to begin with, all the filaments are bunched up and crashing into each other. The rowers can't get a good grip. If the muscle is stretched out too far, the myosin heads can't even reach the actin rope. But at that optimal length, you have the maximum possible number of myosin heads able to grab and pull on actin. More connections means more force. Simple as that.
All right, we have our engine. We know how it works. But how do we turn the key in the ignition? That is what excitation-contraction coupling is all about. It's the critical process that links the electrical signal from your nerve, the excitation, to the physical sliding of the filaments, the contraction.
Pay close attention to the timing on this graph because the order of events is crucial. First, you see that sharp spike? That's the electrical signal, the action potential. It happens almost instantly. Immediately after, you get this huge, rapid release of calcium inside the cell. And only after the calcium shows up does the muscle actually start to generate force or tension. Remember the sequence: electric spark, then calcium release, then muscle contraction. Always in that order.
And here it is, the whole shebang in one beautiful flowchart. Seriously, this is gold. If you're going to screenshot one thing from this whole talk, make it this one. It walks you all the way from the nerve signal to the release of calcium from its storage tank, the sarcoplasmic reticulum, to that calcium binding troponin C, which, as we know, finally lets the cross-bridge cycle begin. This is your study guide on a single slide.
So we've pretty much mastered skeletal muscle. Now we're going to switch gears a little and talk about smooth muscle. That's the muscle in your gut, your blood vessels, the stuff that works without you thinking about it. Some of the principles are the same, but the differences are what examiners love to ask about.
Okay, this is probably the single biggest difference you need to burn into your brain. Look on the left. In skeletal muscle, we know calcium's dance partner is troponin. Easy enough. But now look at the right. In smooth muscle, there is no troponin. It's just not there. Instead, calcium binds to a different protein called calmodulin. This then activates an enzyme, myosin light chain kinase, or MLCK, and that's what switches on the myosin. It's a completely different on switch.
This table is your perfect cheat sheet. Skeletal muscle? It's striped. You control it consciously, and its calcium sensor is troponin. Smooth muscle? No stripes. It's involuntary, and its calcium sensor is calmodulin. And one other key difference to note. Smooth muscle gets calcium from both its internal storage and from outside the cell, while skeletal muscle relies almost totally on its internal stat.
And one last little detail on smooth muscle. It basically comes in two flavors. You've got unitary smooth muscle, like in your intestines, where all the cells are linked together and contract as one big sheet. Then you have multi-unit smooth muscle, like in the iris of your eye, where each cell can be controlled individually. That allows for really fine, precise movements.
Okay, let's lock in everything we've learned with a quick knowledge check. Take a look at the question on the screen. Which of these statements about skeletal muscle are true? Go ahead and pause if you want a second to think about it.
So, how did you do? Little bit of a trick question. They are all true. Statement A is the heart of the sliding filament theory. B is the fundamental action of the cross-bridge cycle. And C is the main takeaway from that length tension graph. If you nailed all three, you are in fantastic shape.
And that's really it. From the structure of the sarcomere, to the cross-bridge cycle, to the different calcium switches, it all comes back to how those filaments slide. If you can master these core ideas we just walked through, you're going to walk into that exam feeling confident and ready to go. Good luck.